So you will see in the website and also now in the screencast how to run the assembly and how to run the annotation. But both process takes a very long time, so there is the pre-calculated output right in our machine. So I log in again. By default we are in our home directory, very good. We have our data sets in data, and in particular for the annotation and the assembly I decided to use the mock community. That is a very good candidate because it's composed by genes that are already fully sequenced and finished. There are very good reasons to use this dataset. But what we are now interested in is to see which kind of output files we get. So first, one of the programs that we can use for the metagenome assembly is Spades. Spades.py will print us a brief manual with the instructions. So we need to use -o to specify the output directory while all the files will be stored. We have to specify -1 for the first parent and -2 for the second parent, and the most important is -meta to specify that this is a metagenomic sample, and so it's important not to assume that there is a uniformity of coverage throughout the whole assembly. That is the default for most of the assembly programs. Finally, we can specify the number of processor with -t or -- threads. So, just to refresh the fact that we can work from different positions, so in this case I will go straight into data and then into mock and in vitro. Okay, I'm here. This is my position. This is what I have in the directory. I have some reads. I have already some contigs produced with another assembler and an annotation. So let's run the assembly with Spades. Then my output, I want to be in the home directory. So I specify tilde, that stands for my home directory, and Spades. Then -1 reads. The first pair, -2, again reads. And the second pair, meta is the most important, and finally -t4 to specify to use 4 threads. Ready? Run. If you try it later with your virtual machine, it will take it about 3 hours, so don't expect a fast process. Let's now have a look at the output of the assembly, the contigs. So, I decided to save the output in my home. In Spades, you will see in the note that there is an assembly elsewhere in the data directory, but you can just follow the notes without hurdles. So, yeah, this is the multi-FASTA file with the contigs. Each config has a name, like node 1, 2, and 3. There is the length in the name and the coverage. That is the chimer coverage. It's not totally overlapping with the sequence coverage. It's a slightly different measure, especially when the chimer size used for the assembly is quite different from the read length. So it can be used in relative terms to see the average, the main difference from a config to the other. So yeah, this config is quite long indeed, but how can we see the statistics of this file without scrolling all the contigs, of course? We already introduced this program, SeqKit. SeqKit prints some average metrics for the sequences contained into FASTA files. So, for example, we know that totally we have 66,000 contigs, and totally we did assemble 50 megabases. The length of the contigs spans from 65, so very short, to the biggest, that was the first that we saw, almost 200 KB. Well, the problem with the distribution of sizes in contig dataset is that there is a long tail of very short contigs, so the average is not so meaningful. It's very difficult to compare two different assemblies just looking at the average. So there is a metric that with Illumina sequencing it's still very used, that is the N50. To enable the N50 calculation, we have to add the --all switch after the stats. So scikit stats --all, and then again the file we want to calculate the statistics for. So the N50 is 1.3 KB. What does this mean? N50 means that if our total dataset spans 50 megabases, half of it, so 25 megabases, are stored into contigs longer than the N50. So there are contigs longer than 1.3 KB, and totally the contigs longer than 1.3 KB account for 25 megabases. The logic behind this metric is to be independent by the from the long tail of short contigs. So we can run the annotation process. Again, you will see the nodes to perform the full annotation. The program that we can use is Proca. Proca, again invoked without arguments, will list all the switches and arguments that we can use. Generally speaking, requires at least one argument, that is the contigs file that we want to annotate. Then we can specify the output directory, the file name of the output. We can specify multiple CPUs, and again, there is a switch specific for highly fragmented genomes. So we can start typing the program. Parameter CPU for -out. Again, I can save the annotation in a proc directory in my home. We can specify a prefix like annotation or ANN. What is important to add at the end is the set of contigs to annotate. So while the program works, and remember it takes about 1 hour and a half with our machines, let's remember that Proca is actually a pipeline. So Proca is able to predict coding genes, rRNAs, tRNAs, and CRISPR, because it's basically a pipeline composed by several programs independently written. So there is Prodigal to detect the coding potential, so the CDS. And after detecting which possible genes will code for protein, the output, so the unnamed protein, is blasted again database of proteins to detect similarities with existing protein. There is another algorithm called Infernal to predict non-coding RNAs. There is RNAmer to predict rRNAs, so ribosomal RNAs, and finally Aragon, that using the anticodon will predict and will annotate the tRNAs. So all of this is running behind the scenes when we invoke Proca. What is the output like? There are many formats. One of the standards is GFF3. That is a TSV, tab-separated values, having these columns. The first is the contig name, the second is the source, that is the algorithm. For CDS, we have Prodigal, but as I said, there are different algorithms predicting different features. Then we have the starting and ending coordinates, we have the strand, the plus or minus, the frame if the case, and the extended annotation. So, if we have matches with a known protein, instead of the laconic hypothetical protein, we have a more complex things, like we get an ID, we inherit the EC number from an enzyme, for example, we have the similarity with existing genes, and the description of the product. So, remember that you have already the annotation in the annotation subdirectory. As you can see, there are many different file types produced. In particular, GFF is the one we saw right now, but there is also the multifast with all the protein encoded, or the genes, so the nucleic acid sequence for each gene. So, let's have a look at the GFF file with less minus capital S to prevent the word wrap and so see the whole file as it is. At the beginning we have header lines. They start with the hash symbol or the sharp symbol. To search for things within less, we can use the forward slash. So let's look for CDS, for example, and then enter. Now we are in the middle of the file. As you can see, a lot of products will just be ORFs with an annotation as hypothetical proteins. Unfortunately, some other protein will have a good match with a databank, so we have lucky examples as well. But as we said, it's not only CDS that are the products of Prodigal. We can also look for rRNA. Let's have a look at the 16S if we find one. Yes, we found one. So, when we find something interesting in this file, what can we do? We can take note of the ID, because this table only contains the coordinates and the annotation. But we can use the ID to then query the FASTA file. So in this case, for the 16S, we have to have a look at the nucleic acid format, not the protein. So we can have a look at the annotation file ending with FFN, that is the FASTA nucleic acid format. So forward slash, let's paste the product, and we have one ribosomal RNA. It's possibly longer than this, but for the moment we can just copy a fraction, just to have a look at what's the output of a BLAST search. Let's paste this. We can also subselect the 16S reference database in this case. The first match is Alovivax rubber. That is hopefully one of the strains that is indeed present in the mock community. So this prediction has been quite accurate. And now it's your turn. You can query the GFF file in a different way than proposed from the notes. You can select some genes that you already know and you can understand better. So good work.